Hey guys, my name is Abhishek and welcome back to MBBS Strategy channel. Today we are going to read about the sensory analyzer pathway of sound analyzer. Okay, so let's begin our today's topic. So friends, in this video we are going to talk about the actual process of sound perception. How the sound stimuli is going to be received by the hair cells that are present in the organ of corti. Okay, so this is the picture that here you can see. This is the picture inside the cochlear duct. In my previous video, I have already explained what are the structures that are present inside the cochlear duct. Let's recap those structures. This is the spiral limbus or the bony projections that comes from the modulus, and attached to it we have the walls of the cochlear duct. Like this is the vestibular membrane or otherwise known as Reisner's membrane, and below or the inferior wall of the cochlear duct is made up of basilar membrane, and surrounding the cochlear duct we have the spiral ligament and the stria vascularis that continuously produces the endolymph. Here you can see this is the tunnel of corti or the canal of corti, and to the inside of the canal of corti or to the medial portion of the canal of corti we have the inner hair cells, and to the exterior part of the canal of corti we have outer hair cells. And over these hair cells we have a thin lamina, that is reticular lamina, and connecting to reticular lamina or over reticular lamina we have a structure or we have a membrane that connects with this spiral limbus, that is tectorial membrane. Okay, so these are the structures that we are going to see inside the cochlear duct. Now we are going to learn that how the sound stimuli is going to stimulate these hair cells, and that will result in generation of action potential in this cochlear nerve. Okay, so let's begin with the function of inner hair cell. These inner hair cells are the primary receptor cells that contributes to 95% of generation of action potential in the cochlear nerve, whereas 5% is contributed by these outer hair cells. Okay, so the inner hair cells plays most of the roles for generation of action potential or for receiving the sound stimuli, and also helps in responding to the sound stimuli. So what happens, this inner hair cell, we have a special structures inside the inner hair cells. What are these? These are stereocilia and kinocilia. Okay, so the finger-like projections that are facing upward in this columnar hair cells is known as stereocilia, and the longest projection or the longest finger-like projections, that is known as kinocilium. Okay, in between this stereocilia and kinocilia we have special structures that connects these stereocilia channels. Okay, these are the special channels that provides the entry of potassium ions, and connecting to it we have special circular or special proteins that connects these channels. What are these? These are tipling proteins. Okay, so the tipling proteins connect the channels of each stereocilia, and this helps in shearing of the hair cells. So the tipling proteins can be cadherin 23 and protocadherin 15. Okay, these are the special type of gene proteins that are formed or that forms these tipling proteins. This connects the channels of different stereocilia. What is the help of this? Okay, so let's start how this sound stimuli comes. First of all, when the sound wave comes to our external pinna, then it beat to the tympanic membrane, and that leads to vibration of the ear ossicles, and those vibrations leads to production of the mechanical energy. Then the ear ossicles, the steps or the foot plate of the step beats over the surface of oval window, and it produces the fluid vibration in the perilymph of scala vestibuli. Inside the cochlear duct, when this perilymph of scala vestibuli will vibrate, then it will also lead to vibration of this vestibular membrane, and then further it will vibrate the lymph or the endolymph that is present inside this cochlear duct, that will further lead to vibration of this basilar membrane. Okay, so when the vibration will come in this whole cochlear duct, then the tectorial membrane, the reticular lamina will also vibrate. That leads to rushing of the endolymph inside this space in between the reticular lamina and the hair cells. So the hair cells is going to shear, and this shearing will generate the action potential. How? When the vibration of basilar membrane will take place due to the movement of endolymph fluid, then what will happen? It will stretch the reticular lamina. Okay, when the fluid will enter in between this space, then it is going to stretch the reticular lamina, and then the hair cells will move. Okay, due to the movement or spontaneous or rapid flushing of the endolymph in between these spaces, it will tend to move the hair cells. Okay, the hair cell is going to shear, and it will lead to the movement of each stereocilia towards the kinocilium. Okay, so the movement of each stereocilia towards the kinocilium will take place. It means that this stereocilia is going to move towards the next stereocilia, and the next stereocilia is going to move towards the next stereocilia. It means the whole stereocilia is going to move towards the kinocilium. Okay, so the due to the movement of this stereocilia and kinocilia, what will happen? The tipilings or the tiplings that are present over the surface or that connects the different stereocilia is going to open. Okay, it stretches the tiplings and it opens the channel for ions. Okay, for what ions? It means that the cochlear duct, or inside the cochlear duct we have endolymph, and I have said that endolymph is rich in potassium ion. So it will open the channel for entry of the potassium ions. Along with the potassium ion, we have calcium ion. So there will be entry of potassium and calcium ion through these channels. What will happen? Due to the entry of potassium ion, it is going to depolarize the hair cells, and due to the entry of calcium ion, it is going to stimulate the synaptovesicles to fuse with the basilar membrane. What do you mean by this? That when the potassium and calcium ion is going to enter through these channels, then then inside this hair cell the potential will increase, and due to increasing of the potential, it will lead to depolarization of the hair cell. And when the calcium will enter, the synaptic vesicles containing the neurotransmitter inside this hair cell is going to fuse with the basilar membrane, and after fusion it is going to release the neurotransmitter. So the next process will be, it will release the primary neurotransmitter. What is that? Yes, it is glutamate. The glutamate is going to be released near the receptor of the spiral ganglion, receptor of the peripheral process of the spiral ganglion. Then the production of action potential takes place at this reception region. Due to reception of the glutamate, there will be generation of action potential at the peripheral process of this nerve fiber, and the information will be going towards the spiral ganglion. Okay, so this is the function of the inner hair cell. Next, what is the function of outer hair cell? As I have said that the inner hair cells contribute to 95% of action potential generated through this cochlear nerve, then the 5% is going to be contributed by this. But this 5% will not matter in receiving the stimuli. Rather, these outer hair cells have more helpful property. What are they? They are the sound modifier, amplifier or moderator, and protective mechanism. They have the protective mechanism. How? These cells, these outer hair cells have more efferent fiber rather than having less afferent fiber. It means the information will be sent, there will be more information sending from this olivocochlear bundle. Okay, there is a olivocochlear bundle just before the receptors of this connecting to the spiral ganglion. Okay, so this olivocochlear bundle is going to send more informations about the actual sound analyzer, how the sound is going to be received by this inner hair cell. It means that if the hair cell is going to be producing more action potential or generating more action potential, or the hair cell will be producing the stimuli to loud sound or to low sound. Okay, so the outer hair cell is going to regulate those sound. It is going to moderate, or it is going to modulate, it is going to amplify, and it is going to protect the inner hair cell. How? In the structure of outer hair cell, we have a critically sensitive hearing protein that is known as prestin protein. Okay, this is only expressed in the outer hair cell, not expressed in the inner hair cell. So this prestin protein that are present in the structure or in the outer surface of this outer hair cell is going to modulate or is going to help in the process of sound modification and protective mechanism. How? This prestin protein is going to relax and contract, and due to contraction and relaxation, the size of this outer hair cell is going to change. Due to relaxation of this prestin protein, it will increase or it will elongate the size of the outer hair cell, and the relaxation of the prestin protein occurs when there will be loud sound stimuli. This will result in elongation of the outer hair cell, and when there will be elongation of outer hair cell, then the basilar membrane is going to move to downward direction. When it will elongate, then it will push the basilar membrane in downward direction. Due to the pushing of a basilar membrane to downward direction, what will happen? There will be decreasing in the hair cell activation. It means that it limits the basilar membrane movement, and when it limits the movements of the basilar membrane, then it is going to decrease the inner hair cell activation. When the prestin protein is going to contract due to low sound stimuli, it means that when the sound is not heard properly, or when we cannot able to discriminate which sounds are coming, then at that moment it is going to contract, and this contracts leads to shortening of this outer hair cell. And when this outer hair cell is going to shorten, it means that the attached or the lower surface attached with the basilar membrane is going to lift up. That leads to upward movement of the basilar membrane. When the basilar movement is going to move up, it means that it is going to stimulate more movement of the basilar membrane, and when there will be more movement of basilar membrane, it means more hair cell activation will be takes place. The inner hair cell will be activating more and more, and it will lead to more action potential generation. Okay, previously when there will be hair cell inactivation or decreasing in the hair cell activation at loud sound, it means that it protects the inner hair cell, and when there will be low sound, then there will be stimuli from this outer hair cell to produce more and more action potential to discriminate which sounds are coming. Okay, so these are the functions of these outer hair cells. Next we are going to read about the ascending auditory pathway, or how the sound is going to travel from this cochlea or from this inner ear to the auditory cortex that is present in the brain. Okay, so the main auditory nerve or the cochlear nerve starts from this peripheral process, right from the glutamate. After when the glutamate is released, then the action potential is going to be generated from the peripheral process of the spiral ganglion. Okay, the organ of cortex will be responsible for production of the action potential in the spiral ganglion. This is the inner ear and this is the outer ear. From the inner ear we have the small structure here, it represents the spiral ganglion, and from the spiral ganglion we have this structure, this is cochlear nerve. So the sound information is going to travel from the spiral ganglion to the next structure that is present in the medulla oblongata through this cochlear nerve. So what is the next structure where the next synapse, the nuclear fibers is going to make? This neuron fiber is going to make the next synapse at the medulla oblongata at this cochlear nucleus. Okay, so we have two cochlear nucleus in each side, and total 4 cochlear nucleus, that is 2 ventral cochlear nucleus on both sides and 2 dorsal cochlear nucleus on both sides. Okay, so the cochlear nerve is going to make the synapse at this cochlear nucleus. Then the information travels from the ipsilateral side of the cochlear nucleus to contralateral side of the next structure in the medulla oblongata. What is that? It is superior olivary complex. Okay, so the left side, or if I am going to say that it is the left side ear, the left side ear is going to send the information through the cochlear nucleus to the contralateral side of the superior olivary complex. So the neuron fibers or the neuronal fibers is going to make the synapse at this complex. The superior olivary complex is responsible for a reflexive localization of the sound waves or the sound information. Then from the superior olivary complex of the medulla oblongata through the lateral lemniscus, the sound information is going to travel from this structure to the next structure that is present in the midbrain, that is inferior colliculus. Okay, so the next synapse that is going to form at the inferior colliculus. Inferior colliculus is responsible for combination of auditory and visual. It is having the auditory and visual location information. It is going to gather both the auditory as well as visual information. From the midbrain or from the inferior colliculus, next the nerve fiber is going to make at the medial geniculate body of the thalamus. Listen, the information travels from the left ear towards the right side of the brainstem. It means that in the direction of contralateral side. From the cochlear nucleus, it is going to innervate the contralateral superior olivary complex, and from the superior olivary complex it travels to the lateral lemniscus to the inferior colliculus on the contralateral side, and exactly at this inferior colliculus some are going to cross, some, but majorly the primary pathway represents this red line and the secondary pathway represents these blue lines. Okay, so the most of the neuronal fibers remains at the same side, means it remains at the contralateral side in respect to the inner ear, okay, from which the sound informations are actually coming. So the primary pathway remains on the contralateral side and it next forms the synapse at the medial geniculate body of this thalamus, and from the thalamus through the internal capsule it is going to innervate the auditory cortex, and this auditory cortex of the cerebral cortex is present at the lateral sulcus of the cerebrum. Okay, so this is the major pathway through which it is the left ear is going to innervate, the left ear is going to send the informations to the right auditory cortex. This is the primary pathway, and also along with that some information is going to travel through the secondary pathway to the left auditory cortex. So we have some advantage due to this both passages of the informations to both right and left auditory cortex, is that when there will be some kinds of pathology in the left ear, obviously the right ear is going to receive the sound, then the right ear is going to provide the informations to the left cortex, and some minor information is going to be traveled to the right auditory cortex along with the left auditory cortex. Okay, so when there will be pathology at left, as it provides the informations to the right auditory cortex, so it will be receiving less amount of information, but the right ear is functioning well, so it will provide much more amount of information to the left auditory cortex. but along with that it is going to provide some information like in the secondary pathway to the right auditory cortex. Okay, so this was all about our today's topic. If you find this video is helpful for you, please do like, share and subscribe my channel. Thank you so much for watching my videos.